Okay, so welcome everyone, the students, faculty, friends, family. I've never seen so many people in the same defense. It's great everyone came. So Niema is a student in Bioinformatics program, like a couple of you guys. Let's see the website. So bioinformatics students have to do a rotation. He approached me, did a quick rotation in my lab, worked on the tree with 1 million leaves on it. And I read about anyone who can work with a tree with 1 million leaves has the technical skills. So I was happy with what was happening. Then he approached me later to ask to work in my lab for his PhD. And if I believed in luck, I would say he's a very lucky person because 5 minutes into, not because he approached me, but because 5 minutes before or after, like into the meeting or before the meeting, I don't remember, I got an email saying that I got my first grant as a faculty. And the grant was on HIV. HIV is, Joel, one of the committee members, had introduced me to this faculty. The only condition for the grant was you should know nothing about the field. And I very easily satisfied that requirement. I didn't know anything about HIV. I still don't know a lot. So I told Niema, look, I just got this grant. If you are happy to work on something where I don't know anything, I will be learning with yourself, go for it. And he did. And this past 3 years, we have been learning about HIV together. We have made some nice contributions you will learn about. At this point, with everything I know about HIV, he knows as well, or at least that's the idea. I will be putting that proposition to test later today, once you all leave the room. In the meantime, he has also been doing a lot of great work on teaching. And you will hear about that from Paula. Just so that everyone knows the way it's going to work, Niema will give his talk. We don't ask a lot of questions, maybe like small clarification questions if you have those during the talk. And then at the end of the talk, you will be asked, you can ask questions. Then everyone leaves the room except the committee. We ask 4 questions, and we'll take it from there. So now Paula will say a little bit about what Niema has done during these 4 years on the teaching side.
So it's rather unusual when the student has 2 advisors on completely unrelated topics. And it creates an impression that Niema was so unhinged. One advisor was not sufficient, and 2 advisors were needed to put him on the right. That is only partially true. Allow me to explain. So Niema is an exceptionally talented educator, much better educator than me, most other professors here or elsewhere. And he could just make a great career by simply being a great educator. But he was so dissatisfied with the current status quo of traditional classroom education that he instead decided to try to disrupt the system, and I believe we will hear many more great things about him as he continues to dismantle the existing status quo. So I knew from the very beginning that Niema was an exceptional educator, and I actually graduated another student who was exceptional educator 5 years ago, who is now a teaching professor at Carnegie Mellon. And I made a mistake that I didn't want to repeat with Niema. I was both educational advisor and research advisor for the student. And these 2 directions sometimes were getting in the conflict because sometimes education was overshadowing research and vice versa, I don't know. Anyway, I decided to avoid this mistake, and I'm very grateful actually that Niema was wise enough to take 2 advisors, and there was absolutely no conflict to complete, and you will see it hopefully from the presentation, that in this case there was no overshadowing and we were very balanced parts.
Thank you. Thanks so much. So thanks everyone for coming. So yeah, thank you all for coming. I know it's late. Hopefully you can stay here for a little bit, but I'll be presenting today. My talk's called Scalable Methods for Molecular Epidemiology of Rapidly Evolving Pathogens. So just a brief overview. These are the sections of my talk, and then I'll introduce each section with some quote that I feel represents that topic. So first, I have an introduction, and being an evolution person, I have to have a Charles Darwin quote, otherwise you don't get your Ph.D. So it is not the strongest of the species that survives, nor the most intelligent that survives. It is the one that is most adaptable to change. So some brief background, a phylogeny, it's a tree structure that shows the evolutionary relationship between different species. At the leaves, we have the present-day species. The internal nodes represent ancestors that we generally assume are extinct. And the distances along the tree represent some unit of evolutionary time. It could be years, it could be generations, it could be mutations, but some metric of evolutionary time. Even though we typically think of them as species, like trees of species, we can also think of them as trees of samples of the same species. So with HIV, this becomes especially important because we could have an independent sample from different patients, we could have samples from the same patient. But yeah, we need these phylogenies that show the relationships between the HIV samples. In the work that I'll be doing, I won't be talking about one of my contributions too much, but it basically set the foundation for a lot of the other things I was able to computationally perform. Basically, with these HIV trees, we get trees of thousands, millions of taxa, and they're very slow to do computation on if you don't have a very fast tool. So I developed TreeSwift, which is a Python package for handling trees. It's currently under review. And just to show some comparisons of why this is important, basically, let me get my highlighter out. Laser pointer. So I'm showing some benchmarking results between TreeSwift and some other existing toolkits, and you'll see TreeSwift is much faster for loading the phylogeny. It's this bottom curve. It's much faster for doing typical operations that you would do with a phylogeny. And not just as far as speed, but in terms of memory usage. Here's a quote from Fast and Furious franchise. This time it ain't just about being fast. It's also a much lower memory footprint compared to the existing tools. So this tool was basically what enabled all the future analyses I'm going to talk about. Okay, so now to try to switch gears a little bit, let's talk about HIV specifically. So an epidemic, it's the spread of a virus across different people in a population. And in a computational framework, we can think of this as we have a contact network, which is a graph, where nodes represent individuals and edges represent risky contacts across which the virus can spread. And then we have a transmission network, which is a subgraph of the contact network. Now it's directed, and again, nodes represent individuals, but edges actually represent transmission events. So each edge is labeled with what time the transmission event occurred. And for HIV, we assume that this graph is a tree. If we zoom in on one specific transmission event, we can see that from a phylogenetic framework, we have the host individual who has an entire population of viral species within their body. At the time of transmission, a single viral lineage gets transmitted to the recipient. I say single because we typically assume that the transmission event's a bottleneck. This could be relaxed, but for the purposes of this talk, just assume that one lineage gets transmitted. And then as time goes on, the virus continues to evolve in both hosts now. When we sequence individuals, we're essentially taking random lineages from each individual and observing the tree of the full phylogeny constrained to just those individuals. And then we can think of evolution of some ancestral sequence evolving over time to produce the sequences that we see in present day. So just to kind of summarize this process, we can think of this generative process as we have a contact network, and then dependent upon that is a transmission network, dependent upon that is a viral phylogeny, and dependent on that are the viral sequences. So we can think of the stochastic process, this is a stochastic process where each step is dependent on the previous steps. In other words, this is a complex conditional distribution dependent on the previous steps. So one contribution I'll be talking about is Favites, a simulation framework I developed to be able to simulate epidemics, all four components of this, to hopefully emulate reality as well as possible. I'll also be talking about a new model of phylogenetic evolution that I had developed throughout my PhD, just to kind of better represent this stage of the process. And in addition to this forward process, in reality, all we can confidently observe are the viral sequences. So there's this problem of, can we somehow infer properties of an epidemic just from the sequences? This initial step of going from viral sequences to viral phylogeny is fairly well studied, so I won't be talking about that in this talk, but we can assume that people at least are confident in their abilities to infer phylogenetic relationships from HIV sequences. But this next step of, given the viral phylogeny, can I somehow infer properties of the transmission network? This is an open problem that is not very well understood, but there's been recent research going into it. I will not even be talking about this. What I'll be talking about is, can we actually circumvent this transmission network? Can we see, well, what are the actual actionable predictions that I can make? And can I make those predictions purely based on the phylogeny? So not necessarily worry about who infected whom, but instead find information that can help public health officials act on the epidemic. And I'll be talking about this as well. It's a tool I developed called Proact, which is currently under review. So the first section of my talk, I'll be talking about the dual birth model, and the quote I like is, "Essentially, all models are wrong, but some are useful," by George Box. So again, I'm zooming in just on the viral phylogeny step. So evolutionary models of phylogenetic trees, they describe probability distributions over the space of trees. And I want to kind of make it clear that the random variable here is over trees. So the tree topology and the tree branch lengths. We can sample from these distributions to simulate phylogenetic trees. So existing work, there's a simple model called the Yule model in which every lineage, the splitting of a given lineage is a Poisson process with some rate lambda across the entire tree. Therefore, the time at which the first splitting event occurs is an exponential random variable with that same rate lambda. The time at which the next splitting event occurs is the minimum of 2 exponential random variables, therefore an exponential random variable with 2 lambda, so on and so forth. And typically, it's nice to use this model because it's very simple. However, with HIV, one thing that I noticed is that the phylogenies that we get in the Yule model, they typically are much more balanced than the real phylogenies that we observe from HIV. So I was asking the question, can I come up with a new model that extends on the Yule model, but allows me to very fine-tune measure the balance of the tree? So I'll be introducing the dual birth model. I won't talk about it too much in this talk because I've already talked about it at previous talks. But basically, it's an extension of the Yule model where instead of having one global rate across the tree, each branch is labeled either active, which is shown in blue, or inactive, which is shown in red. And the active lineages split with this rate lambda b for birth rate, and the inactive lineages split with this rate lambda a, which is activation rate. And lambda b should be greater than or equal to lambda a. When they're equal, that reduces to the Yule model. So just to briefly summarize what I did with that work, just because I won't go into depth, I developed a linear time algorithm to sample trees under the model. I derived the following useful mathematical properties. The expected number of cherries, where a cherry is defined as an internal node that has 2 leaf children. I assume that all trees simulated on the model are binary. The expected number of active leaves, the expected branch length, and the expected pendant branch length. And in addition to these mathematical properties, I use these properties to develop 2 methods to estimate the model parameters of a given tree. And in terms of biological results, I inferred a phylogeny of close to 1 million ALU transposable elements. And just to give a little bit of background for people who aren't familiar, ALU elements are transposable elements that occur in all primates. The human genome has close to 1 million of them, and their function is not very well understood. But basically, it's hypothesized that some of them are active, some of them are inactive, but the proportion of them that are active is an area of debate in the literature. So using my methods under the model, I actually was able to estimate the proportion of active ALU elements, which is very close to the estimates that are currently thought as what is accurate in the literature. And I was able to estimate the rates of ALU activation and propagation, which I've estimated here. So I just want to, the actual values themselves aren't important, but note that under the Yule model, these 2 rates would be equal. But I was able to infer rates that the activation rate was 2 orders of magnitude smaller than the birth rate. So these were somewhat interesting results. I'd be happy to answer questions about this offline, but for the sake of the talk, I'll be moving on. So the next section of the talk is Favites. It's the framework that I developed for simulating epidemics. And the quote that I like is, "A big computer, a complex algorithm, and a long time does not equal science." And just some background information in this, I had seen a lot of simulation studies that tried to do some form of inference based on simulated epidemics, but they didn't do a very careful job designing the experiments in terms of model selection. And really, if you do a simulation experiment, making sure that your models are appropriate is essential to making sure that the results that you get are at all meaningful. So I developed this framework called Favites, where it's a flexible framework that allows the user to choose what specific model assumptions they want to make at every given stage of the epidemic. And to show this, for this figure, don't really worry about understanding the specifics. The main thing I want to point out is that I've taken the entire epidemic simulation framework, and I've basically broken it down into these independent, not necessarily independent, but these modules that are abstract modules where a given implementation of the module defines the model assumptions that you make. And in this way, a researcher can basically plug and play specific module implementations at each step to pick whatever assumptions work best for them. So some important features to highlight: it's completely modular. So again, users can plug and play the models and model parameters they see fit. It's easy to develop plugins, so you can either write plugins from scratch in Python 3, or you can actually wrap around existing tools and just put them into the framework. And I place an emphasis on portability and usability, so I have a Docker image that sets up all the requirements for you. In addition to developing the tool, I also performed a simulation experiment in which I was trying to emulate HIV in San Diego between 2005 to 2014. I used model parameters that we learned from literature and from real data sets, and I'm just going to go over some of the most important ones for the relevance of the talk. So this is the transmission model that we used. It's a continuous time Markov chain in which we have different states: susceptible, which is S; acute HIV untreated, which is AU; acute HIV treated, which is AT; chronic HIV untreated, which is CU; and chronic HIV treated, which is CT. And again, this is a continuous time Markov chain where a given individual transitions between these states with some given rate. So between the infected states, all of the rates are constant, and then transitioning from susceptible to acute untreated, so your initial infection time, is the sum of all of your partner's infectiousness rates. So if you don't understand the specifics of this, don't worry about it too much, but basically we have these 5 states and we learn their relative parameters from literature and from real data. So in addition to the transmission network, for the contact network, we chose the Barabasi-Albert model. Under this model, contact networks, the graphs that you generate have, they're scale-free, so they have power law degree distribution, which is representative of the sexual contact networks that we typically see with HIV. We also tested 2 other models, the Erdos-Renyi model, which is a simple model in which you basically flip a coin for each possible edge, as well as the Watt-Strogatz model, which generates, it basically generates graphs that have small world properties. So it gets a lot of small subcommunities within the graph. For sequence evolution, I used the general time reversible, or GTR model, with gamma rate heterogeneity. And I tried to emulate HIV-1 subtype B, and specifically the pol, or the polymerase region of the virus. And for tree evolution, I used a coalescent model in which we had bottlenecks at each transmission event, and the viral population experienced logistic growth. And as far as sampling, I sampled all individuals at the very end. And by sampling, I mean it's equivalent to them coming to the clinic and getting sequenced. So I did a 10-year simulation, and I sampled everyone at the very end. This is unrealistic, and I'll talk about later in the talk a more relaxed version of this. But for the sake of this specific part of the talk, assume all individuals were sampled at the very end. So we did this simulation experiment, and the first question that someone should ask is, do the data that I get look realistic? So most tools that analyze HIV data, they look at pairwise distances. So I thought, well, maybe I should look at the pairwise distance distribution. So basically, each one of these violins is the pairwise distance distribution of the sequence that you get. These green ones show real data. I have these ones are San Diego, and these right 2 are a simulation to emulate Uganda, but I'm only going to focus on San Diego in this talk. And basically, we see that in the default parameter settings, which I show in purple, we get a distance distribution that looks very similar to the real San Diego distribution. And I'm not showing right now, but I have supplemental materials if you want, but there's a kind of double peak phenomenon that you typically observe with real data, where you have 2 peaks of really short distances, one of direct related transmission events, and one of just somewhat closely related transmission events, and we do observe those in our simulated data. So we at least feel somewhat confident that, at least based on pairwise distances, our data look realistic. Using our data sets, we can perform some type of studying of the real world, where I can basically change parameters of my models and see how does the outcome of the epidemic change as a result. So on the horizontal axis, I'm showing expected time to begin antiretroviral therapy. This is a treatment in which, as long as someone adheres to the treatment and becomes virally suppressed, they essentially become non-transmittable. So to the right, I'm showing you're starting treatment faster. So 1 means on average it takes 1 year to start treatment. This means on average it takes half a year, a quarter of a year, so on and so forth. And on the vertical axis, I'm showing the ratio of untreated versus treated individuals in the population. And as you'll see, no matter which model of contact network I choose, we get fairly consistent results. And at this line, I'm showing when untreated equals treated, so we have equal number of untreated and treated individuals. And UNAIDS basically is a United Nations group that they have this 90-90-90 goal where they want by 2020, 90% of people who have HIV to be diagnosed, 90% of those people to be on treatment, and 90% of those people to be virally suppressed. And we'll see that in San Diego, we're between 1.5 a year on average, which is right around this goal. So it shows that this goal is actually potentially a realistic goal as long as we can get the rest of the world to the same level of treatment that we get in San Diego. So these are some interesting results that could theoretically be used in a cost-benefit analysis or some type of proposal for public health. And in addition to studying the epidemic, I wanted to benchmark existing tools that try to analyze HIV data. So typically when people obtain HIV sequences, they do what's called transmission clustering, where they try to cluster individuals based on similarity of their viral sequences. So some tools that exist to do this, HIV trace is a common one. Joel is a big part of developing this tool. Cluster Picker is a tool that so HIV trace uses sequences, Cluster Picker and Tree Cluster use phylogenies. Tree Cluster was built in our lab. And basically, these are different approaches that have different objective functions of clustering, but the question is, how well do they work with respect to what people want to do? That question is not such a trivial question. How do I define how well does something work? Each of these tools has different objective functions, so there's no ground truth that I can compare all of them on. So what I thought instead was, what do we use these tools for? Generally, people measure the growth of clusters over time and use these tools to try to do surveillance and see which groups of individuals are more rapidly spreading the virus, and then try to predict who we can intervene so that we minimize the spread of the virus. So instead of accuracy, I wanted to look at effectiveness. Using each tool in whatever framework that they advertise of being able to predict future transmissions, or at least predict something about public health, let's see which one does it best. So specifically, I inferred transmission clusters at year 8 of the simulations. I measured the growth from year 8 to year 9. And the methods of how I did this, it's in a paper that was published in Journal of Infectious Diseases in 2018. And then I averaged the number of transmissions in the top 1,000 individuals each method picked to see which one was the most predictive of future transmissions. And you can get a plot that looks like this. So on the horizontal axis, again, I have whatever variable that I'm changing. In this case, it's expected time to begin antiretroviral therapy. So as we go to the right, we're getting faster starting treatment. And on the vertical axis, I'm showing what is the average number of infections of those 1,000 people that you chose at time 9, between times 9 and 10. And we see, and then on the bottom plot, I'm showing what you would expect if you just randomly picked 1,000 people. And then I'm showing what you would get using Tree Cluster and using HIV trace to do your clustering. And we see that, as we would hope, both methods do significantly better than random chance, so this is nice. We can even study how well do they work as we change whatever parameters of our epidemic. So the previous plot is now this one, where now every plot I'm changing the expected number of sexual contacts in the network. So in this example, that was the previous figure. On average, each person had 4 sexual partners. Here, on average, people have 2, 8, and 16. And we can see the trends of these tools as we vary these parameters. And still consistently, both methods do better than you would expect by random chance. Yeah, so that's all I'll talk about for that project. And then the next project I had, it's Proact, and the quote that I like for this is, "It is difficult to make predictions, especially about the future." So as I mentioned, we might not even care about doing transmission clustering per se. It's a useful technique, but what I really personally care about is, what are some actionable pieces of information we can obtain, and can we obtain that information directly from the viral phylogeny? So some background information, there's generally there's two ways of preventing the spread of the virus effectively. You can either target people who are currently living with HIV and try to prevent them from transmitting to other people, for example, antiretroviral therapy treatment, or you can target people who are not infected and try to prevent them from getting the virus. For example, pre-exposure prophylaxis. In my talk, I'll be focusing on this top scenario. How do we take people that are currently living with HIV and try to predict which ones are most at risk of spreading the virus, and how can we ensure that those individuals stay on antiretroviral therapy? So to try to answer this question, the current state of the art is we should prioritize based on growth of their respective transmission clusters. So as I mentioned, we can cluster the viral sequences, where the intuition here is that closely related viruses imply a recent common source of infection. Then we can measure the growth of those clusters over time, and we can prioritize members of the fastest growing clusters. That's exactly what I did in the previous section. But my question is, well, if we're given a phylogeny, by doing this transmission clustering, even though it's an effective way of summarizing the data, we still are summarizing the data, and we could potentially be losing useful information. So is there any way to do this type of prioritization directly on the phylogeny? So my goal is, ideally, my input would be a rooted phylogeny T with branch lengths, and my desired output would be some ordering of the leaves of T, ideally in descending order of number of future transmissions. So this is the mindset that I want to work towards. And some key insights to motivate the method that I developed. Let's imagine the simple scenario where person A infects person B at time t1, and then later person A infects person C at time t2. If you imagine what the phylogeny would look like, we basically have, this is all of the viral lineages within person A, this is within person B, this is within person C. At each transmission event, so T1 and T2, we have a bottleneck event. And then TA, TB, and TC are the times that these three individuals got sampled. And you'll notice that basically, if we imagine the viral phylogeny at time TB, so only including person A and only including person B, we had some phylogeny of just these two individuals, and they each had some incident branch length. And then at time TC, when we observed the sample from individual C, their sample coalesced with A, and then their common ancestor later coalesces with B. So it's equivalent to placing C on this incident branch length. And as a result, the incident branch length of A is going to decrease. Another scenario would be, what if C coalesces with B first? So in this scenario, I have C coalescing with B, and then their common ancestor coalesces with A. And this is what the phylogeny would look like. We had some phylogeny at time TB, the same exact one as before. But now, when I sample person C, I place their sample on the incident edge of B. And as a result, person B's incident branch length shortens. It turns out that this first scenario is more likely than the second scenario. And you can read about some simulation studies that went into figuring this out in the citation over here. But basically, what I'm getting at is this top scenario where person A, the person who's transmitting, their branch length shortens is much more likely than the other two scenarios. But a given branch length will never increase. It will only shorten. So in simulations, I wanted to see how often does this hold up. My intuition was, theoretically, I should have a correlation between the number of, or an anti-correlation between number of transmissions and your incident branch length. And each person's branch length should decrease over time. So in this plot, I have on the horizontal axis normalized time since infection. So 0 means the moment you got infected, and 1 means the end time of the simulation. And then on the vertical axis, I'm showing average incident branch length of whichever individuals fall into each of these categories. So this line, each x-axis point, so let's say this is 0.4. This means that 40% into their time of infection, this is the average incident branch length of all individuals that had 0 transmissions, and so on and so forth. And as we see, the higher transmission individuals do indeed, on average, have shorter incident branch lengths. So this motivates a very simple approach we can take, which I call Proact, prioritization using ancestral edge lengths, where the input, as I would hope, is a rooted phylogeny T with branch lengths. And now just a very trivial algorithm, let's just sort the leaves in descending order of incident branch length. So the highest priority individual has the shortest incident branch length. If two individuals are tied, let me break the tie using their parents' incident branch length. If they tie again, use the grandparents' incident branch length, so on and so forth. And if one of the two individuals that I'm comparing runs out of ancestors, but I had ties the whole way up, just use its average branch length from it to the root of the tree in place of an ancestral branch length. Okay, so this is the algorithm that I came up with to do prioritization. So now we want to see how well does this work. Again, I did a simulation experiment where the transmission model I used was the exact same as before, but then I made slight changes to the other simulation parameters with respect to the previous section. And I'm going to highlight those changes. So with respect to the contact network, I'm still using the Barabasi-Albert model, which again, the graphs that you generate are scale-free. But now I'm enforcing communities of densely connected communities. So specifically, I'm making 20 Barabasi-Albert communities, each one with 5,000 individuals. And on average, a given person has 10 edges with people within their community, and on average, 1 edge outside of their community. And then sampling, so as I mentioned before, I was previously sampling everyone at the very end of the simulation, but this is extremely unrealistic. This would be as if all humans all get sequenced at the very exact same time. Instead, what's more likely is that people will get sampled the first time they begin antiretroviral therapy, which is standard in many developing countries, and it's standard in San Diego, which is what I was emulating. Okay, so in simulations, I can come up with figures like this, where the horizontal axis is, let me look at the proportion of individuals that I've selected in my prioritization technique. So this shows, let me pick the top 10% of the sample population. This is, let me pick the top 8%, etc. And then the vertical axis shows, in the selection that I made, what is the average number of transmissions that those people made? So essentially, you can think of this as a cumulative moving average, where this is the average of the first 2%, the first 4%, the first 6%, so on. And then I show Proact, and I show cluster growth. Where cluster growth, this is the same method I described before, where you measure the growth of the clusters from time 8 to time 9, and then prioritize in descending order of cluster growth. And we see that Proact consistently outperforms the cluster growth approach. One question I had is, well, is this sensitive to subsampling? That could potentially mess things up. Okay, sorry.
Can you describe the y-axis?
Sure, yeah, sorry. So for any given selection of individuals, the y-axis is showing what is, oh, sorry, yeah, I got to clarify. So the y-axis, it's not showing exactly the number of transmissions. This is an adjusted value where, so I've taken the number of transmissions on average of my selection, and I've adjusted it such that 0 is what I would expect by random chance, and 1, which isn't shown in this figure, is what you would get with the optimal ordering. Sorry, good question. Yeah, so if that doesn't make sense, feel free to ask any more clarification questions. But yeah, so this is adjusted, so 0 is random, 1 is optimal. And yeah, Proact consistently outperforms cluster growth. And then I wanted to see, well, how sensitive is this approach to subsampling? So for each of these two approaches, I then randomly subsampled the full data set. So 25% means I just randomly picked 25% of sampled individuals, etc. And then I performed the methods on what I pick. And we see that the two methods, they stay roughly similar performance no matter how I subsample, at least based on these values. Note that this is still adjusted transmissions per person. So one depicts the average number of transmissions in the optimal ordering of the top 25% of people that I picked, and the 50% of people I picked, so on and so forth. And note that the horizontal axis is a proportion with respect to how many people I had sampled. So this is 10% of the 25% of people that I picked for this point. But yeah, in general, we see that still this conclusion that I got, that Proact is outperforming cluster growth, it seems to remain consistent as we change, as we subsample. So then again, I picked simulation parameters, but those parameters might not be accurate. So I wanted to see how sensitive is this to the parameters that I chose. So the figure that I had shown previously is this left figure. And then first I wanted to ask, well, how does it change as I vary the rate at which people start treatment? So as I go to the right, I'm doubling the speed at which people start treatment. So I'm having the expected time people start treatment. So this is, on average, people start within half a year, or in other words, the rate is 2 inverse year. And on average, people start treatment in a quarter of a year. And still, we see that the results seem consistent. And we actually, yeah, it seems Proact is consistently doing better than we would expect by random chance, and it's consistently doing better than cluster growth. Next, I wanted to see, well, what if I change the rate at which people stop treatment? So I want people to adhere to treatment, but you'll always have people who stop. Again, the left plot is showing the default that I had shown previously. And then now I'm speeding up the rate at which people stop treatment. Or in other words, as I go to the right, people are getting worse at staying on treatment. And you see that, again, the results that I got seem consistent, and it actually seems like we're extending the gap between cluster growth and Proact. And similarly, if we decrease the rate at which people stop treatment, so in other words, as we go to the right, we're getting better at keeping people on treatment, we see that it still seems like Proact is generally doing better than cluster growth, but they overlap significantly when we get to the best possible scenario of keeping people on treatment very well. Okay. So in addition to changing the rate at which people start or stop treatment, I wanted to see, well, what if I change how connected the graph is? So again, the left one was my default, where on average, individuals have 10 sexual partners per person. But then if I say 20 sexual partners or 30 sexual partners, how does this change? And again, we see that the results look consistent, and the difference between cluster growth and Proact is clear. Okay, so this was all on simulated data, but I wanted to see how well did I do on real data? Because really, if it doesn't work on real data, then it's not as important to me. So in simulations, my metric of how well I did, I know exactly who infected whom, so I can measure my efficacy based on how many people were infected by the individuals that I chose. With real data, though, I don't know who infected whom, so I need to use some form of a proxy that I can observe. And I chose to use inferred relationships, such as HIV trace links. So HIV trace defines two people as genetically linked if their pairwise viral distance is within 1.5%. So the phylogenetic distance between their viral sequences. So I wanted to define links using multiple approaches. So one is what I just described. 1.5% means you're linked. And if you're over 1.5%, you're not linked. But if you imagine this is somewhat of a strict scoring function where if two people are 1.5% different, they are linked. And if they're even 1.51%, they're not linked at all, which is equivalent to if they're 50%, they're equally not linked. So I wanted to have a more smooth scoring function as well. So I used two theoretical smoother scoring functions and one that I empirically learned, which I'll describe in more detail in a second. So first, these are the two theoretical scoring functions I came up with, and this is the Proact just default step function at 1.5%. We see it has a step function at 1.5. But then I have two sigmoid functions that they have their point of inflection at 1.5%, but one of them is steeper than the other. And you can read the manuscript to get the exact functions that I use, but basically they're smoother and softer than the step function. So in addition to these two theoretical functions that I'm just kind of arbitrarily picking, I wanted something that is actually learned from real data. So I came up with this empirical score function that I learned from real data, where the motivation here is I imagine that the pairwise distance distribution is a mixture model of three roughly Gaussian distributions, where the first distribution is pairwise distances between two individuals that had the same recent source of infection. Then we have a second, slightly larger distribution of people who are within the same subtype, and then a larger distribution between people who are the same subtype. And my score that I chose, given some distance, is the value of that distance given the probability density function of the first Gaussian divided by the sum of its probability density function value for all three Gaussians that I learned. And the actual score function that I used, you can see here. It's a little further to the right than the other ones, which is why I wanted to keep the other two examples as well, but this one was completely learned from real data. Okay, so now let's actually test using these score functions. So I use an HIV dataset of 926 HIV1 subtype B whole sequences that were collected in San Diego from 1996 to 2018. These are the same sequences I used to learn that empirical score function. To benchmark, I divided these into deciles. So the first 10 percentile, 20 percentile, sorted in chronological order of when they were obtained, where each decile essentially defines two sets: pre, which is all sequences obtained before that, up to that decile, and post, which is all sequences that were obtained after that decile. And then for each decile, I performed prioritization using all of the sequences in pre, and then I measured my efficacy by measuring number of links in post. And then to measure how well I did, well, I can measure. So I can just sort these individuals in descending order of the scoring function to get an optimal scoring. And then I can use the Kendall-Tau correlation coefficient to see how well does my ordering correlate with the optimal possible ordering using that scoring function. So just some background information. Kendall's Tau, it's a statistic measuring the ordinal association between two lists of numbers, and the values range between -1 and 1, where 1 means that there's perfect agreement, -1 means there's perfect inversion, so complete anti-correlation, and 0 means there's no association. And specifically, I use tau b, which adjusts for ties. And this is, let me just describe this plot for a little bit. So each of these horizontal axis points is one of those deciles. So this is the first decile, second decile, third decile. The vertical axis is showing Kendall's tau. I'm showing the null distribution in gray, where to sample this null distribution, I just randomly shuffled the order a bunch of times. And then the blue points are what you get with the Proact prioritization, and the yellow points are what you get using genetic distance with cluster growth. And I've labeled anything that was not statistically significant with an exclamation point, where I defined statistical significance using 0.05 alpha, but with Bonferroni multiple hypothesis test correction. So we see that when we use the step function that was the strict step function at 1.5%, cluster growth is outperforming Proact, which it's somewhat expected because this is essentially the prioritization that cluster growth is doing. right? Cluster growth, you're inferring clusters with HIV trace, which defines these clusters using 1.5% as its threshold. And then you're predicting which ones are going to grow the fastest using the same exact scoring function. So even though this is interesting because we are still doing prediction of the future, it's not completely unprecedented that it would do better than Proact. But let's see how it works as we use the different scoring functions as well. So using the stricter sigmoid functions, this was the steeper but still smooth sigmoid function, we see that Proact does similarly to cluster growth for a lot of these. So in some data sets it does better. For some data sets, cluster growth does better. I mean percentiles, not data sets. And we see that basically both tools are doing much better than you would expect by random chance. And in this example, none of them were, so all of them were statistically significant. If I use the smoother step function, so this one is the less steep but still sigmoid theoretical function, now Proact is fairly consistently outperforming cluster growth, but cluster growth is still fairly consistently outperforming random, except for this one example where we fail to reject the null hypothesis. And then with the empirical smooth step function, so again, this is the one that I learned from the 3 Gaussian mixture model, we see that Proact consistently outperforms cluster growth except for this one point, and we have these 2 points where cluster growth was not statistically significant. And I just want to emphasize that this is very interesting because it shows that even with just 10% of the data set, we have fairly high predictive power with this metric of efficacy. Yeah, so this was the last slide I had on this section, but I want to just give some lasting discussion on Proact. As you noticed, the algorithm that I came up with is extremely simple. It's just sorting the leaves by incident branch length. And I'm hoping that in addition to providing this method to public health officials, this kind of lays the framework for how we can kind of explore these questions in a formal computational framework. So I've come up with a way of actually performing simulation as well as real experiments to see how well a new method can do. And I'm hoping in the future, so I used incident branch length, but there might be other properties of phylogeny that could be also predictive or maybe even more predictive. So I leave that to future work. Okay, so that was the last research-focused section of my talk. And then I want to end it with my contributions to education. And the quote that I like is, "Learning is not the product of teaching, learning is the product of the activity of learners," by John Holt. As Pavel mentioned, I've been trying to kind of break ground in the traditional way that classes are taught, and I've mainly been involved with developing these massive adaptive interactive texts, which for those of you who don't know, they're essentially online texts with auto-graded challenges embedded throughout them that help promote an active learning style of education. So what does this acronym mean? They're interactive, so learners directly interact with the text and solve challenges as they're learning. They don't wait till the end to be able to solve challenges. So for example, here's a code challenge within one of my texts where they implement this algorithm pattern count. This is actually from Pavel's online course that we've developed, where this is an algorithm where they're counting the occurrences of a substring pattern within a string text, and we gave them pseudocode. And then they basically are given this coding box that they can just do directly within their web browser without any need to worry about set up anything on their own machines. It's adaptive, so it can actually give them uniquely tailored feedback to their specific misconceptions. So in this example, I gave them pseudocode, but let's pretend that we have this student response. And for people that know Python, you'll notice at the end of it, in the pseudocode, you go up to the length of text minus length of pattern inclusive, but in Python, range is exclusive at the end, so the bug is right here. They have an off by 1. And my autograder can actually detect this and hint to them that, hey, you're failing to account for an occurrence of pattern at the end of text. They're massive, so they can support ultra-large numbers of learners. So these numbers, I got them, I think, last week. I have roughly 40,000 students in my own data structures text that I've developed. And in the Bioinformatics Algorithms Part 1 text that I've created with Pavel and Philip Compeau, we have roughly 35,000 individuals. If you want to learn more about my philosophy between how I develop these courses and how I, my pedagogy, you can learn about my teaching philosophy in this TEDx talk that I gave, I think in 2017. But I want to just quickly summarize some of the specific contributions I've made to bioinformatics education. First, with Pavel and Philip Compeau, so Pavel Pevsner and Philip Compeau, I developed this Introduction to Genomic Data Science course, which is mainly meant for people that have some programming background and some computer science background to learn the bioinformatics, the algorithms that are kind of the backbone behind common bioinformatics analyses. However, you do need programming background for this code. course. So I wanted to also develop a course that I call Analyze Your Genome, again with Pavel and Philip, that it's now catered towards biologists. So instead of teaching you the nitty-gritty algorithms and mathematical theory behind these tools, let me just teach you how do I use these tools and how do I interpret the results, how do I interpret where error can come from, and how do I make sense of these without necessarily having a super formal computational background. Separately, on my own, I've developed the Data Structures and Active Learning Approach text. So this one, it's been wrapped around a course on edX, and it's also been being used at UCSD as well as University of San Diego and University of Puerto Rico in their data structures in-person courses. And I've since adapted it to a physical textbook that's available on Amazon. And yeah, so those are kind of my primary teaching contributions. And I want to end with a summary, and I thought the most fitting quote for the summary would be from The Return of the King. It's done. Yes, Mr. Frodo, it's over now. So I first talked about Favites, which is the simulation framework that I developed. It's been published in Bioinformatics in 2018. I talked about the dual birth model, which is a phylogenetic model of evolution that specifically I was trying to be able to fine-tune how balanced the trees that I simulate are. I developed Proact, which is a prioritization technique that uses phylogenies of viral sequences. It's currently under review. And I developed TreeSwift that I didn't really talk about into detail, but I'd be happy to talk about it offline. Ultra-fast Python package for doing tree manipulations and tree traversals. It's also currently under review. And as far as bioinformatics and computer science MOOCs, I've developed multiple courses on edX and Coursera between 2017 and now. And I wrote a textbook. It's on Amazon. And I want to just end the talk with some acknowledgments to some people that have made this possible. The quote, this is my favorite quote ever. Folk in these stories had a lot of chances of turning back, only they didn't because they were holding on to something. What are we holding on to, Sam? That there's some good in this world, Mr. Frodo, and it's worth fighting for. So a Ph.D. is a very tiring and time-consuming process, but you just kind of keep on fighting for the people that you're helping and for the people that are supporting you along the way. So first, I want to acknowledge my funding sources because I got to eat, and they help me, they help me survive, literally. I want to most importantly thank my Ph.D. committee, Siavash, for being a great research mentor. I've asked him like so many, I'm sure he thought very dumb questions along the way. I knew nothing about phylogenetics before joining his lab, but he was patient with me the whole time. Pavel for taking me under his wing, teaching me what is bioinformatics education, how can we shake the world. Vineet Bafna couldn't make it today, but he was mentoring me ever since undergrad, and he's been always kind of like this sage source of wisdom for any questions I have about academia, as well as about career options, life. Joel taught me literally everything I know about HIV. I'm sure I asked a lot of really dumb questions at the beginning then too. As Siavash mentioned, neither of us knew anything. So anything that sounded somewhat intelligent, it's because Joel taught it to me. Davey, he also couldn't be here, but he was also one of my main sources of kind of wisdom on the HIV front. I want to thank my collaborators. So Liz was my co-author on the data structures textbook. Manon was one of Joel's postdocs at the time, and she helped me develop Favites. Christine, Mia, and Leo, who Mia is here, they've been kind of mentoring me on the education front throughout the years and helped pave the way for me to start a teaching faculty position in this department starting in like a week, I think. My lab mates, who a lot of them came today, without their support, without their help in getting things running, just asking for advice, things like that, there's no way I would have been able to make it. My PhD friends that I've made along the way, this is one of my favorite pictures of the whole squad, but yeah, such great memories that we've made over the years, and I'm truly blessed to have such a good group of friends. My high school friends, who I still hang out with all the time, these are my best friends, Felix and Ryan, that I don't think they could have made it today, but they work, unfortunately. Yeah, I mean, they still support me all the time. And then couldn't do it without my family. My uncle and aunt are in the audience. My mom and dad and sister and brother-in-law are in the audience as well. Yeah, thank you all for your support. And I'm happy to take any questions right now.
Huh? Can you go back?
Oh, yeah.
Okay, so I must say this. I had not noticed this is a slide. Otherwise, I would have corrected him saying that there is still 5 to 10 minutes before the lecture. Hopefully, it's not done at this time. So we will figure that out now by asking questions, and then the committee will have the meeting. Please.
Sure, Owen.
I have questions on Proact evaluations when we go back to those evaluation graphs.
Yeah, sure. Real data or simulated data?
I think either or.
Like these ones?
Sure, we'll start with that. So question one would be, so in all of those simulations you show, your cluster growth thing is basically random? Is that expected? What does it mean that this cluster growth metric that people use is basically equivalent to paying people at random?
So it's consistently a little better than random, but yeah, I mean, I think it depends on how you define. Well, yeah. So on simulated data, pretty much it looks that way. I think part of it is the fact that the simulations aren't completely representative of reality. I would want to. So the models that I chose happen to be models that have already been developed and well studied. I had a couple preprints that I was working on where I've been trying to come up with, like, how do we come up with better contact networks? How do we come up with better transmission model, things like that. And I'm expecting that if you do better model fitting, like more appropriate models, it should do better. But yeah, based on these results, that's what it looks like. And yeah, you can. Yeah.
Okay, so now going to real data.
Real data.
Yeah, and so this part, I think I just need clarification.
Sure.
Going on, but normally when we train on a larger set, we expect to see performance increase, and in all of our plots here, we see that the 10% is the one that's actually the most performance.
So yeah, that's a great question. So basically the question was, usually when we train, if we had more data, we usually see an increase in our predictability. Why don't we necessarily see that here? So keep in mind that I'm measuring, so I'm doing my prioritization on everyone up until this decile, and then I'm ordering the people afterwards. In this example, I'm only ordering based on 10% of the people, and a lot of those people just didn't infect anyone. So you have a lot of ties of like 0 or like score of 0 in this example, and as a result, you would expect that you wouldn't have as good a predictive ability just in this metric, like based on this specific metric. So I would imagine if I had a different metric, or if I just always did it based on the 50 percentile, I might see cleaner results. But yeah, I'm not here. I get to see 90% of the people in my post set, where here I only get to see 10% of the people. So I would assume part of it's because of that. Yeah, Ben.
So, I mean, I want to start off by saying, just commending you on, taking on this, what seems like an exceptional challenge of, looking at phylogeny in this high level, back in the space, and thinking, what looks like a really impactful problem.
Thanks.
And, yeah, just great job with that. But I'm really interested in this space in particular, and I'm curious if you have done made any effort to characterize how sparse the evolutionary space is. I know it's a small genome, I know it's a compact genome, but how likely is convergence, and how much does that limit your ability to make statements about how the evolution is happening?
Yeah. Okay. So background for other people in the audience. So with HIV, you see, or it's hypothesized that you see convergent evolution, where because of the high selection pressure, if there's like one sequence that's most fit, you will see the different viral populations converge to it. In these samples, I was looking at the pole region, which is also under high selection pressure. If I'm not mistaken, which I would have to double check, I think most of the selection pressure happens within host, but I guess it could happen across host. But I didn't consider those in this talk, but that would be interesting. And I think it would cause my distances to be slightly less than what the true evolutionary distances are, because we'd have multiple mutations that look invisible to us because they converge to the same thing. But that's a great question. I haven't explored it, but that'd be good for future work. Bill, and then Patrick.
I think kind of connected to that. You had that slide where you had A, B, and C, and you showed the two different ways that they could converge. Just kind of clarifying. It makes sense, I guess, when you had the A one, yeah. So the top one kind of makes sense because they both split off from A. Is the bottom one only possible with convergent evolution, where they have to get together?
No. So convergent evolution is with respect to fitness, and it's typically, like, I would imagine primarily with the sequence. In this case, it's just by chance A has a lot of viral populations, and by chance it happened to be. So if you think about it in the reverse time coalescent theory, it just happens that we don't have a coalescent event, don't have a coalescent event, don't have a coalescent event. Oh, now when all three lineages exist, we all of a sudden have a coalescent event by chance.
So it just happened to get a very similar one when it was transmitted to like the earlier, like both transitions happened with very similar strains. It wasn't a more evolved strain from time passing?
Yeah, it had to, like, the strain that went into this individual, like, or was the ancestor of this individual, and the strain that was the ancestor of this individual had to have been very similar because they had to have had occurred very very high in the tree. Yeah, great question. Pat?
So on the real data, you're trying to identify clusters which are most likely to spread the disease, right? Which clusters are most likely?
So I'm trying to find individuals, specifically individuals.
Okay.
But currently, what most people do is they do this transmission clustering, and then they measure how do those clusters grow over time. And then individuals that are within the fastest growing clusters, those might be the most risky ones.
So I have two questions there. One, can you correlate your outcome with real clinical predictors? Somebody comes into a clinic, is diagnosed with certain demographics. Does the hospital have a risk chart where they say this individual is at a high risk of continuing to spread, so you have to be especially active? And can you overlay that with your predictions, which is sequence-based?
Yeah, so that's a great question. So I specifically actually, maybe I should have emphasized this. I tried to avoid any demographic information. We actually had some demographic data for the real people. So this is a paper that Joel's lab wrote last year. They actually did a predictive model that incorporated, so they had their just purely sequence data-based predictions, and they also had a model that they trained using like metadata from the patients. I wanted to avoid any metadata just to avoid any potential bias that I think when you're dealing with something sensitive like HIV, unnecessary bias could have like potentially bad repercussions. But I could imagine you could incorporate that type of data as well. And they do have predictors that use patient data. Yeah.
Second question there, since you have time course data, did you see a change in the number of edges and connections between the early time points and now with apps such as Tinder and so forth going on, where instead of being just local, you can start connecting with anybody random wherever you are? That's
a great question. I think that also ties into Owen's earlier question regarding limitations of the models. So no one asked about it, so luckily no one put me on the spot, but I'll just reveal myself. All the contact networks that we had were all static. So over the 10-year period, they did not change their sexual contacts, which is extremely unrealistic. And specifically, as you mentioned, in this window of time, we have like these online apps for finding people to have relations with. I'm assuming you would need some type of dynamic model. How that would look, I don't know. I do know that I have room in Favites to be able to implement those, but I just don't have the models. No one's done the math to see what is a good model, how do we sample this model, so on and so forth. But that would be a great question to explore in the future. Yeah. Yeah, Jeff?
I guess that's kind of what I was interested in, like the big level. We talk about the contact network and then transition network, and then all the way to the sequence level. But then in terms of evaluating it, it seems like you didn't really have data, I guess, on the contact network and the transition network, but like, is there a way you can incorporate that, or is there data that you could use to kind of evaluate how well the model you're creating are doing?
Yeah, so in with HIV, I know you can have patients answer survey data that they can list individuals that they think they might be connected to. And there has been research that's gone into prediction of directionality of transmission events. I chose to stay away from that because I think it's not very well understood, and you could use my tool to try to gauge how well those tools work. So actually we had a student group, I think Jonathan's group. Did you guys do direction of infections? But in Siavash's class, we actually had a group that tried to explore how do we measure the accuracy of these different techniques. But that's a great question. I just chose not to go that direction. Any other questions from the audience?
Okay, so we'll have the audience step out, and then they can be asked questions.